chorda mesodermal cells push towards the anterior portion of the animal. So in mammals and reptiles and birds, we expect that as the notochord is developed from chorda mesodermal cells, as it elongates anteriorly, the primitive streak also regresses in terms of its length. No? The chorda mesodermal tissue that is underlying the ectoderm actually is an important factor because it appears that the inductive activity it does, or it's been shown that the inductive activity it possesses, alright, is influenced by two processes: trans induction or vertical induction and planar induction. So in trans induction, right, or vertical induction, the chorda mesodermal cells or notochordal cells that serves as the primary inductor produces inductive signal which will be perceived by the outer ectoderm, and if the outer ectoderm perceiving the signal is competent enough to interpret it, then it will proceed with neurulation. On the other hand, planar induction happens along with the whole or happens in the competent ectoderm, that if a certain portion of the competent ectoderm um, is activated by the inductive signal coming from the mesoderm, coming from the notochord, then it the activation spreads in a horizontal axis, no, or in a planar direction. So from one cell to another cell of the ectoderm that lies on the same plane. Now, how important is the chorda mesodermal tissue or the notochordal tissue in the process of neurulation? Basic question we have to ask, or we have to know the answer, is in the absence of the chorda mesoderm or the notochord, what will happen to the ectoderm? A few experiments, grafting experiments in particular, have have proven the importance of the notochordal tissue in the process of neurulation. That in its absence, in the event that the chorda mesoderm notochord is absent or has been removed or excised from the growing embryo, the ectoderm underlying it is not going to develop as neural ectoderm. No neural tube will develop. In other setups in which a competent ectoderm that already interpreted the inductive signal, or that already received the inductive signal from the chorda mesoderm, has been excised from a post-gastrulation embryo. Okay, on such setups, it's been shown that the excised ectoderm continued to develop as neural parts, as neural tubes, so suggesting that the whole process of primary neural induction is requiring the presence of the tissue. Without it, no primary neural induction will take place. Okay, so on the macroscopic or gross perspective, we're now seeing how neurulation is accomplished by our general ectoderm. Okay. Um, on the cellular perspective, we can appreciate, or we can describe the process of neurulation to actually start with some changes on the morphology of the cells that are targeted, or the cells that are targeted by our inductive signal coming from the chorda mesoderm, coming from the notochord. These cells manifest a change in their shape, right? So we know that